Okay, so good afternoon, everybody. Thank you for joining our today's webinar, which is entitled Development of Sara-home, a novel assessment tool for patients with ataxia. It will be presented by Gessica Vasco and Susanna Summa from the Ospedale Pediatrico Bambino Gesù in Rome, Italy. My name is Anja Hermans and I work at the University Hospital Tübingen in Germany at the Institute of Medical Genetics. I'm responsible for the training activities in the European Reference Network for Rare Neurological Diseases. And this webinar is part of a joint webinar series which we organized together with another European reference network, the one for neuromuscular diseases, and with the European Academy of Neurology. And I'm very happy to have today such a topic which covers actually both of our foci. We have or three even. We have a webinar which has a specific disease or covering ataxia. Then it has a focus on neurorehabilitation and has a pediatric focus. Okay, so a couple of words for those of you who are from the other part of the world, so not from the Europe, on the European reference networks. So the European Reference Network for Rare Neurological Diseases is one of the 24 ERNs in Europe, which have been established by the European Commission in 2017. The goal of these networks is to collaborate better, to intensify their exchange of knowledge, because they are groupings of expert centers, and by networking they can achieve this better exchange of knowledge and experiences, which would be of final benefit, of course, for the patients that should receive a state-of-the-art quality of care. So our network has at the moment 32 full members institutions, which are in the dark blue countries, and 10 affiliated partners in the light blue countries here on the map. And we are expecting that other approximately 30 institutions join our network in 2021. And we are covering 6 disease groups that you can see here. So some general information about the webinars. As I said, we are focusing on rare neurological, neuromuscular, and movement disorders, and have particular focus also on neurorehabilitation or on pediatric subjects. Normally we have 40 to 45 minutes of presentation, and at the end we have 15 minutes time for questions and answers. This webinar is being recorded and will be published on our website at the latest 2 weeks after today. And you may want to check other parts of our website, for example, the Disease Knowledge Hub on Ataxia, which compiles useful information on this disease. And your browser will automatically open a post-webinar satisfaction survey at the end of the webinar, where you can write your impression, you can suggest topics for future webinars, and just tell us your opinion, which we appreciate a lot because it helps us to improve the webinars. And even though these webinars are aiming mostly medical personnel, we also have patient representatives. And here I would like to show you a short video from Mary Kearney, our patient representative for the disease group Ataxia and HSP.
My name is Mary Kearney. I come from Ireland. I'm a patient advocate for ataxia, which is part of the Rare Neurological Disease group of the European Reference Network. My job is to make sure that the voice of the patient with ataxia is heard throughout the ERN, and I feel I am well placed to do this as I have 2 children with ataxia. I know the daily needs and challenges that present to those with ataxia, and I feel I can bring this information into the ERN network discussions, activities and decisions. I am a member of Friedreich's Ataxia Research Alliance Ireland and Rare Diseases Ireland. As the aim of the European Reference Network is to share, care and cure rare diseases, I feel it is absolutely imperative that patient advocates, clinicians and researchers work together. I fully support this initiative of specialised training in rare neurological diseases, as I feel the sharing of information in Europe and worldwide will help to improve the care for those with ataxia. And the sharing of information, ideas, research results will help to bring a cure for ataxia to all sooner rather than later. It is my pleasure to meet you all.
Okay, thank you, Mary, for the video, and I hope that you will receive also valuable input from this particular webinar, also for your children. Okay, so we go further, and I'm very pleased to welcome today our 2 speakers, Gessica Vasco and Susanna Summa. Foremost, I like this constellation of having 2 speakers, which one of them is medical doctor and the other bioengineer. So this shows this interesting and meaningful interdisciplinary work and the need for such work and the nice results that are coming from it. Gessica Vasco has obtained her medical degree from the Catholic University in Rome, and since 2013 she is pediatric neurologist at the Children's Hospital Bambino Gesù and the Neurorehabilitation Center in Rome. Her research focus is on neurodegenerative and neuromuscular disorders such as Friedreich's ataxia, early onset ataxia, and Duchenne muscular dystrophy. And she's performing research ranging from bench work until clinical studies. Also, one of her key other activities, she's member of the Natural History European Consortia for Friedreich ataxia, so for translational studies in Friedreich's ataxia. And Susanna Summa is a bioengineer. She has received her PhD from the University of Genoa, and she's now researcher at the same hospital as Gessica, and she's performing movement analysis of pediatric patients with ataxia and human-machine interaction for the assessment of neuromotor diseases and for neuromotor recovery with robotic platforms. So thank you very much, Gessica and Susanna, for being here today, for taking time and sharing your knowledge today with our attendees. I will stop sharing my screen.
Okay.
And yes.
Okay. Okay. Good afternoon to everyone. Thank you all for coming. Thank you, Sandra, for your very kind introduction. Thanks also for the opportunity to participate to this webinar. So I'm going to speak to this topic, development of Sara-home, a novel assessment tool for patients with ataxia. And this would be the outline of this webinar. I will give you a brief introduction, main clinical features about cerebellar ataxia. I'll give you an emphasis on critical issues about ataxia rating scales, And then I give the floor to Susanna Summa. She will present the research project, digital assessment tool that we have done, the Sara-home, and the conclusion and keywords. And okay, this is a preliminary question to know your professional background, just to know what is the professional background of the audience.
Yes, I have started the poll and we will wait some seconds for everybody to be able to respond. Okay, 10 seconds left. Okay, and here are the results.
Okay, here are the results. So we have a lot of physiotherapists, very good. Adult neurologist and child neurologist. Okay, so I'm gonna talk about the ataxia. So for the neurologists, but also for non-neurologists about ataxia. And we can start with a brief introduction. I start with the word ataxia. Actually, it's a Greek word and it means a lack of order. So in term of ataxia is sometimes used to designate cerebellar disease in general. In terms of classification, I will use the same categories which have been presented in the previous webinar on ataxia, so which are no genetic, acute, acquired, or recurrent, and in the other side, in genetic form, we have a progressive one and no progressive one with congenital start. We can also differentiate ataxia in terms of anatomical location. So we can distinguish a cerebellar ataxia, a proprioceptive ataxia with somatosensory system involvement at various levels from peripheral receptor to parietal lobes and dorsal columns, and also a vestibular form, quite rare in children. We can move forward and more in details. The topic of this webinar is how to assess the cerebellar function. First of all, clinical examination. We need a neurological examination and we need to explore motor function related to cerebellar performances. So we start with the gait, and the main feature of gait ataxia is a broad-based gait with the loss of balance, unsteadiness, irregular wide-based gait. We need to look also at the coordination of limbs, and normally in neurological exam, we can perform the task of nose finger and finger nose and heel shin. In cerebellar ataxia, movements of limbs are very irregular, fragmented, and there is often tremor. We have to also look at the balance in stance. So we need to look at stance in normal and in tandem, but also in sitting position to observe possible swings of the trunk. The eye movement are also affected and they appear very interrupted. Saccades are very jerky, interrupted pursuit. The fixation has instability and there are square wave jerks. There is also nystagmus. And the speech is normal slurred and impaired in speed and volume in ataxia. But in a characterization of the clinical phenotype, we must not forget particular aspects which are listed here. In congenital form, in a very early onset ataxia, the clinical phenotype in the first year of age of life is characterized from hypotonia. This hypotonia can be really severe, and sometimes it can be confused with a neuromuscular disorder in the first months of life. Very often there is a motor delay, a speech delay, as well as ocular movements, abnormal ocular movements, and ocular dyspraxia. Also, there is often seizure. But in addition, we do not must forget also in many cases the no cerebellar manifestation like spasticity, peripheral neuropathy, Parkinsonism, dystonia, myoclonus, and other motor signs. But in clinical practice, it is routinely accepted that the clinical assessment of cerebellar disease can be achieved by clinical rating scales. Here I present a list of principal scales assessment divided in rating scales and in functional tests. Some of them are disease specific and has been applied only to a specific disease. There is a difference between rating scale and functional test because initially it was suggested that cerebellar symptom could be better captured by a semi-quantitative rating scales. But later on, quantitative functional measures were introduced in attempt to increase the reliability measure, so functional tests. And now I want to ask you which is, in your opinion, the major challenge in assessing ataxia? So this is the second question for you.
Okay, there was a question if there are multiple answers that could be right. This question is rather a single choice question because of the major challenge. So just please answer what you think is the major challenge. Okay, yeah. I think I can show the results.
Okay. Validation for cerebral disorder, inter-rater reproducibility and reliability for children. Okay. The answer are all true, and in the next slides, I will try to explore more in detail these aspects. And I start with this study. This is a review by a specific task force of MDS, specific on topic of rating scales programs. And they has conducted a systematic literature research of the rating scales and functional tests available. All the identified scales and functional tests were reviewed and ranked by the panel. So they ranked as recommended, as suggested, or only as listed by using established criteria. These criteria considered clinimetric properties of the scale, strength, weakness, and a very deep analysis of the scales. Now, without going into all details, the review concluded that SARA scales and ICARS are the most used ataxia scales in the literature. ICARS has a good responsiveness and did better than SARA in terms of responsiveness in patients with spinocerebellar ataxia and Friedreich ataxia. But for sure, SARA scale offer more advantages in terms of dimensionality and better reproducibility. Also, the shortness of the scale is a very advantage for patient with fatigue and with patient with behavioral comorbidities. In general, we can say that the limitations of the rating scales are very similar, and all rating scales has limitations, and in particular we have to deal with a ceiling effect. That means that the utility of the scale in patient on the more severe stage of the spectrum of the disease is very poor. In general, this is a very big problem already unresolved because patients severe affected are not easy to assess. But at the same time, rating scales suffer from floor effect and are not adequate to assess patients in prodromal stages of the disease or in early onset of the disease. Finally, there is a lack of validation for other cerebellar disorder. Some scales are developed for one cerebellar disease, but there is a poor validation for the other disorders. When we move in pediatric population, there are more problems because children get tired earlier, so fatigue is sometimes a problem. We have also to consider that many of these patients with congenital form present developmental delay, intellectual disability, and therefore it can be very difficult to administer scales. In children, there is another problem, and is the age-related maturation of the nervous system. So sometime we can, this could result in a false ataxia scores in younger children because there is a physiological immature motor features, and this physiological immature motor feature can overlap with ataxic features. So the age validation is a big problem. We know very well that a neurological system has a step of maturation, and we know three principal concurrent neurodevelopmental processes. There is a cerebellum growth and development, but there is also a selective elimination of abundant neuronal connection. There is the process of myelination of the central and the peripheral nervous system. And this is very well known from the anatomical study with MRI. Cerebral growth and development has a delay peak compared with the rest of the brain. And also some region of the cerebellar are different, has a different stage of maturation. So for example, vermis, which is very responsible for axial stability, is completed at the age of 8, more or less, whereas the anterior and superior posterior cerebellar region are stabilized at the age of 14, 17. But we have also studied that investigate in reverse how ataxia rating scales are influenced by age-related development. In this study from Deborah Sival and from the Cerebellar group of the European Pediatric Neurology Society, there is a focus on the age validation of these scales in children. They used ICARS, SARA, BARS, and 9-hole pegboard test. the scales are very correlated each other, and it is well demonstrated that ataxia rating scales are age dependent until the age of 12, more or less. In the comparison of these scales, the Sara scales is the best one because can reach the plateau at the age of 10, but in the other study where there is a big validation of the age validation with a broader group of healthy children, it is very clear that that youngest children has a very different outcomes, and only at the age of 12 they can reach a plateau. So the outcome are similar to what we see with the adult healthy adult study. So we have, this is the study that has a validation of the Sara scale for children, but we should take into account that the variation of developmental score across ages in childhood is an important point, and normal references value are still needed, and more broader study with more children are needed. We have also to consider that many of these patients display developmental delay, intellectual disability, and therefore it can be very difficult to administer scales. Anyway, in clinical practice, Sara scales is largely used and diffused in adults and in pediatric clinical setting. The scale was originally developed for adults, but we have an age validation for children. It is composed of 4 domains: gait, posture, kinetics, and speech. The score vary from 0, no ataxia, to 40, more severe ataxia. The advantages are the shortness of the scale, and also there is the advantages that it excludes oculomotor scores. Another advantage is that the outcomes of the Sara are averaged and less detailed in comparison with ICARS. So, as I said before, there are a lot of advantages, but there are so many questions already open. So the low sensitivity and the poor reliability in very youngest children, and so on. For this reason, in literature, in all movement disorder, there is a new trend and to move faster forward and to switch from semi-quantitative rating scales to quantitative measure with the implementation and with the use of new technologies. So in the last decade, more and more studies have been demonstrated the useful of the application of low-cost, wide accessible technologies like wearable devices or digital interface. So the definition of a new digital biomarker is the new challenge in the field of the outcome measure in neurological disease. In line with this, I and my group are involved from 2017 in this big project. Is a big project on pediatric ataxia and public health, funded from Italian Ministry of Health to realize a digital version. We are involved in work package 3, and the aim of this work package is to realize a digital version of a cerebellar assessment for children using low cost and widely accessible technologies. In this project, we have focused the attention on pediatric ataxia, recruiting healthy children, and 2 categories of cerebellar ataxia: a progressive one like Frigga ataxia, ataxia telangiectasia, Arsax, and other, and another group of chronic or no progressive malformatives cerebellar ataxia, like Joubert syndrome and other no progressive cerebellar ataxia. For what I said before, we have chosen to start from Sara scale. And the aim is to develop the development of a digital version of Sara for the evaluation of patients also in remote monitoring. The other aim of the project is also the development of a system for rehabilitation at home, but this is another focus and is out from this webinar. Everybody knows the importance of telemedicine now more than in the past, also for this pandemic situation. So this is the aim of this study was to broad the implementation, to implement the use of technologies in rehabilitation. So before I leave the floor to Susanna, I want to ask to the participants which, in your opinion, are the beneficial of technologies for the remote assessment? And this is the other poll for you.
Okay, 15 seconds left. You can still vote. Okay, I will show the results.
Okay, all the previous. I'm agree with you. And now I give the floor to Susanna because this is the topic of her talk.
Okay, thank you, Jessica. So, as all of you already said, there is not only an answer to this question, and for sure there are several beneficials that are not listed in this previous slide. But while developing the Sara-home, we paid attention to the objectives of digitalizing the Sara scale, providing a quantitative and objective measure, obtaining a continuous monitoring of the patient, and avoiding uncomfortable situations during the evaluation. Therefore, we developed this Sara-home, that is the automation and digitalization of the Sara using low-cost tracking devices and the digital interface with a software architecture based on web services. And first of all, we try to understand the level of acceptance of patients and of their parents using at home such a novel technology without the support of a clinician or of an operator. We asked to a small group to fill out 2 questionnaires. The Quebec user evaluation of satisfaction with assistive technology, that is a widely used tool for evaluating a person's satisfaction, and the IMI, the intrinsic motivation inventory, that measures the subjective experience of patients while performing a task. In particular, this is a 33 items multidimensional measurement tool, which assesses participants' interest, enjoyment, perceived competence, effort importance, usefulness, and felt pressure and tension while performing the given activity. So with this first study, we understood that there was a high interest and participation from patients and the satisfaction with the perception of ease of use from the parents involved in the assessment. So we have now question 4. That is, which items drives, in your opinion, the SARA progression?
So this is a multiple selection question.
Yeah.
Okay, 15 seconds to go. Almost half of you have already voted. And here are the results.
Gate. Okay, then stance. Okay, good, very good. So we can find the answer to this question thanks to Pandolfo, and who analyzed 54 patients with Friedreich ataxia with the aim of characterizing the pattern of disease progression. So just look at the figure on the right. The first thing to know is that the gait and the stance items are responsible for half of the SARA score progression. But this is true until patients remain ambulatory, as shown in figure A and B. Then look in figure C, we have the only SARA item that is performed without visual control, the heel to shin slide, and you can see that it is uniformly abnormal even in patients with SARA under 10. While with the sitting item, it is often initially normal, in some cases up to a total SARA score of 20, and then it progresses linearly. In fact, it is one of the items contributing to the SARA progression in advanced disease after loss of ambulation, reflecting worsening cerebellar ataxia and truncal weakness in advanced disease. Then dysarthria and upper limb cerebellar ataxia, such as speech, finger chase, and nose to finger items, so figures from E to G, may also be initially absent and show most progression after the loss of ambulation, so in these areas. Finally, the alternating upper limb movement item becomes severely abnormal early due to slowing more than irregularity of movement. Therefore, because of the previous considerations and because of some technology limitations, we developed all the items with the exception of the nose to finger test and the heel shin slide. But also we use the PATA test for evaluating the speech disturbance. And this is an example of our user interface. Okay, this is the Sara-home software architecture. We use the RESTful interface Bifrost that allows the access that executes services such as the generation of the user interface, the data collection, sending data on the server database, and the data mining and analysis. Such a technical slide is only to say that the advent of Internet of Medical Things and of the information communication technology, which allows the remote control of different devices and the real-time communication with clinicians has further revolutionized the traditional care system. Therefore, when we talk about innovative technologies, we have to consider not only the hardware, so the devices, but also the innovation in terms of software and communication. So this is the last question, question 5. Because it is fundamental to choose the right technology. So which would you choose?
So 10 seconds to go. And here are the results.
Wearable device. Well, it depends from how do you define the wearable device. Accelerometer, smartphone. Okay, nice. Yeah, very interesting answers anyway. So let's see. I try to find an answer to this question looking on PubMed, and well, I looked for the number of papers that were in the last 10 years were used with low-cost technologies for two diseases that are Parkinson and Ataxias. As you can see, the number of studies is very high for Parkinson, but this is true in general and not only studying the efficacy of low-cost technologies for both rehabilitation and assessment. Consider that if you use as keyword the Parkinson, you will find 70,000 results, while with Ataxia you will find only 18,000 papers. It is also interesting to notice that the most used device in Parkinson are accelerometer and FOS platform since 2010 that are colored in orange and in purple. And smartphone wearable devices since 2015. So they are colored in pink and wearable device, smartphone, sorry, pink and yellow. We can say that in Ataxia we have a similar trend about the technologies, but of course the numbers are very low with respect to Parkinson. So in our project we choose two markerless motion capture devices, Microsoft Kinect and the Leap Motion Controller. They are both based on the infrared technology, and connecting them to a personal computer, it is possible to acquire biometric data of Sara scale thanks to our digital interface. We choose these two devices because we wanted to record the movement as natural as possible, and therefore our aim became to put in relationship the Kinect Leap Motion data sequences and the clinical Sara scale assessment. Both devices provide a skeleton structure that we use as feedback shown on the screen during the execution of the task. And patients reported as more compliance and motivation in completing the tasks. In these movies, you can see some items acquired as example. Okay. Okay, in order to provide the collaboration of the children and to better allow the assessment in absence of the clinician, we also provide two different types of instructions for each task. The conventional one and a movie where a friendly actor placed into a home context strikes patients' attention without speaking, sort of Teletubbies, and guides the task execution. So the challenge was to acquire objective motor information correlating them with the disease by the Sara scale in order to obtain what we call the digital ataxia markers. And because of our experience in gait analysis, we first validated the digital markers measured during the task of gait with the Kinect and the commercial motion capture system that we have available in our laboratory. We looked for the presence of a linear relationship between the parameters acquired with the two systems in order to validate the Kinect system. And also we looked at the correlation between the measures obtained with the Kinect and the Sara scale and the gait score item also, finding more correlation between the Sara score and the stride length and the average speed of gait. The validation of the system and of the digital markers were also confirmed by the Bland-Altman test, as is reported in this table. And then we tested different classifiers accuracy to recognize 3 levels of disease severity: low, medium, and high, and to discern the healthy controls, the chronic non-progressive ataxias, and the progressive ataxias. We found very good results using a super vector machine classifier of disease severity that is shown in this figure. As you can see, the ellipses is very easy to distinguish the 3 ellipses, so the 3 classes. While in figure B, we are able to distinguish the groups with an accuracy of 70%. So we are now following the same data analysis in all the items of the SARA, proceeding with the feature selection of the most representative digital markers, and then classifying the impairment as low, medium, high, finding different level of accuracy depending on the indicator and on the task. And finally, selecting the indicators of the items most representative of the SARA scale, we are combining them in a SARA classifier with a final accuracy that arrives at 92%. Similar and very interesting projects are these that I'm going to show you in the next slides. This one is the APCO test. That is a smartphone application with which the authors evaluated upper limb ataxia and balance. Both the methodologies and the results are similar with our studies. The authors conclude that the app is an easy, reliable and valid evaluating system to quantify the trunk sway in a static position and during the gait, and to assess the severity of the upper limb ataxia. This other research group assessed the upper limb ataxia test in patients and controls using motion measures obtained from a Kinect camera and wearable motion capture devices on NMU. The combination of multimodal features improved the ability to distinguish between patients and controls and to measure the severity of the upper limb ataxia. However, if you're interested in a systematic review about the usage of such technologies in rehabilitation and assessment of people with ataxia, I suggest you to read this paper, where the final conclusion is that these low-cost and innovative technologies are useful and reliable, but because of the rarity of the disease, it is difficult to design something different from a case control study, as it happens in the previous studies I've just showed you. So in conclusion, well, I hope that has been passed the message that it is necessary to define and standardize the measures in terms of digital biomarkers in addition to validate the technologies. The choice of which technology to use is related to the context in terms of the task and environment. The acceptance and the ease of use of technologies is fundamental for a home-based system, and so far studies are isolated experience with technologies used only by its original developer, while multicentric studies are needed, especially with rare disease. About our project, we are in line with the current methodologies and results obtained looking at the baseline, but moreover, this is a longitudinal observation with 2 follow-up and 20 participants per group. And now you are looking at the sensitivity to change of the Sara-home assessment. Thank you for the attention.
Thank you, Susanna. Thank you, Gessica. And now we can await the questions in the Q&A box. Please write your questions. There is one. I think this came as a first question during the beginning of your talk. What is IMU?
Susanna.
DNR?
IMU.
Oh, it is an IMU. In inertial measurement unit, it is exactly the same of the wearable. Well, now when you read, when you find written in a paper wearable device, you usually they are talking about an IMU. That is a combination of accelerometers, a gyroscope, and magnetometer.
Okay, thank you. We can wait another minute if there are some other questions from the audience. While waiting, just to remind you that we have another webinar next Tuesday on functional movement disorders. So this would be the last webinar in this year. So you're all invited to join and to register as usual through our website. Okay, there is one question now from Alan Thomas, who is a patient himself, as I know. Great presentation. Are there any concerns about adherence with remote assessments?
You want to answer? Or I can say that the assessment, the remote assessment was quite good, but for sure there are some problem at home with some device. So sometimes there is need to have a call with family to assess the device and also to the perform. But generally speaking, the acceptance of the family and of the patient was very good. We have some technical problems. Yes, this is real.
Right. Okay. Are there more questions? Yes. Do you also use spatial temporal parameters to assess the patients?
Me, you, Jessica. Me?
Yes.
The study that I show you about the validation of the Kinect with the motion capture system was exploring the spatiotemporal parameters.
Okay, so yes.
Yes.
Yes, and there is also the comparison of the spatiotemporal parameters between KINET and within gait analysis, which is the gold standard for gait assessment.
Okay. Thank you.
You're welcome.
Okay, so we can wait some 10 or 20 seconds more, and if there are no questions, we can conclude this webinar. Yeah, comment from Alan Thomas. Thank you. I think remote assessments are more acceptable to patients and will give more real-life results.
Yeah. This is what we want to demonstrate or to explore. The big problem, I think, is that we need the community of researchers who focus on these topics need to do something joint, because we need to choose digital biomarkers and to prove the validation in big number and in other laboratories. So far the experience very isolated with only one group, and this is one of the biggest problem, because we need to validate the same biological marker in other condition, in other laboratory, and with other technologies. So there is a lot of work to to do.
How many trials did you use to process the gait parameters in the study?
Well, usually we use 3 trials while performing gait analysis.
Okay. Any other questions? Okay. So there are no more questions. So thank you again to Susanna and Gessica, and to all of you attending this this webinar. There is another comment from Alan Thomas. Please connect with Erin Huff-Davis from Aparito Health, a Welsh company developing wearables.
We did.
Yeah, so
we did, and we are in touch with. And so thanks also Bart for work on this, because we need to join the force and to try to do something similar in with other partners. And this could be very useful for all, I think.
Okay, yeah, great. Thank you, Alan, also for your input on and for trying to to establish the connections.
Yeah.
Okay, so what do you think about the future of video analysis and deep learning? This was a question through the chat.
Okay. About machine learning and video analysis, Aparito is doing something like that. They're using machine learning for the video from the camera of the smartphone. And it is a very, very interesting solution. I don't know.
Okay.
I mean, what I try to say that all the algorithm, the type of data analysis that you can do, and all the technologies that you can use are good depending on the context. So it depends, but they are very interesting from an engineer point of view, of course.
Yeah. Okay, so as I see, there are no more questions. Thank you, everybody, and see you soon. Stay safe and have a nice evening. Bye-bye.
Thank you all.
Thank you.
Thank you.